Okay, let's unpack this. Imagine a silent, invisible enemy that's been challenging human health for, well, over a century, constantly adapting, forcing us to rethink our most fundamental medical strategies.
Yeah, a real hidden adversary.
That's what we're diving into today, the world of pneumococcal infections. You've given us a fantastic stack of sources, including incredibly detailed excerpts from Chapter 146 of a comprehensive medical text.
It's really rich material.
It's packed with everything from the historical discovery of this bacterium to its intricate biological mechanisms, how it spreads through populations, and, the cutting-edge ways we're trying to treat and prevent it.
What's truly fascinating here, I think, is how deeply intertwined our collective health is with these microscopic organisms. This deep dive isn't just about listing facts, it's about connecting the dots,
Right.
allowing us to grasp the complex interplay of history, biology, public health, and medicine through the lens of one incredibly persistent pathogen. We're going to pull out the most important nuggets, those surprising facts, and help you emerge genuinely well-informed without feeling overwhelmed.
Sort of getting that professional level grasp.
Exactly. Think of it as a masterclass, giving you that kind of elite understanding of this critical area.
Exactly. Our mission is to distill this rich information, making sure you understand not just what happened, but why it matters, and, crucially, what's next.
Mm-hmm.
And to really set the stage, our sources open by framing a massive global health challenge, one that sets the context for everything we'll discuss about pneumococcal infections. The escalating crisis of antimicrobial resistance.
Ah, yes, the resistance issue. It's huge.
It's been flagged globally as an urgent threat, a real red flag for public health worldwide.
And that designation of urgent isn't chosen lightly. It really signifies a severe and immediate threat, one where our current tools are failing or, soon will be.
Right.
Our sources explicitly name two prime examples that highlight this urgency alongside the pneumococcal infections we're focusing on. First, pneumococcal infections themselves are deemed urgent because we're seeing them more and more worldwide, and they've developed alarming levels of resistance to multiple drugs.
So our standard treatments aren't working as well.
Exactly. The concerning reality is that there are very few, if any, effective antimicrobials left to treat severe cases. That leaves doctors with incredibly limited options. It's a tough spot.
Definitely. And the second example.
The second explicit example is N. gonorrhea, the bacterium that causes gonorrhea. Its urgency comes not just from its growing resistance, but also from how easily it spreads from person to person, often asymptomatically, combined with an alarmingly small number of effective drugs still available.
A perfect storm, really.
It is. And beyond these, our sources point out that many other common infections are also showing resistance, forcing doctors to use second-line treatments. These alternatives are often less effective, or they come with more severe side effects that patients struggle to tolerate, fundamentally changing how we approach even routine infections.
So it's not just that bacteria are becoming directly resistant to the drugs we use to kill them. Our sources also highlight another urgent concern, Clostridioides difficile, or C. difficile.
C. diff, yes.
Which operates on a slightly different but equally troubling principle.
That's a crucial distinction, yeah. C. difficile disease isn't typically about the bacterium acquiring resistance to antibiotics in the traditional sense, where it genetically changes to evade the drug.
Okay.
Instead, C. difficile infections are intimately linked to antibiotic use itself, because these drugs wipe out the normal beneficial bacteria in our gut, our microbiome.
Right, the collateral damage.
Precisely.
Mm-hmm.
When that healthy balance is disrupted, C. difficile, which is often present but kept in check, seizes the opportunity to flourish. And what makes it particularly urgent is its ability to form spores.
Spores, right, like little survival pods.
Exactly like that. There are super tough, dormant seeds that are incredibly resilient to cleaning products and can easily spread throughout healthcare environments. This poses a persistent, difficult-to-eradicate challenge for infection control.
So if we step back, this truly highlights how broad the impact of antibiotics can be. It extends beyond just creating drug-resistant germs. It can indirectly make us vulnerable to entirely different threats by, messing with our internal ecosystem.
Precisely. It's a testament to the complex ecological web within and around us. Understanding this interconnectedness is vital for fighting back.
Makes sense.
And that's exactly why the CDC, the Centers for Disease Control and Prevention, has emphasized a set of 5 core actions to tackle these urgent problems. These aren't just vague goals, they're specific, actionable strategies.
Okay, what are those 5 core actions? Lay them out for us.
Right. The first is infection prevention and control. This goes beyond just general cleanliness, obviously. It's about implementing evidence-based activities, like rigorously reducing device-related infections in hospitals, things like catheter-associated urinary tract infections or central line-associated bloodstream infections.
But really common hospital-acquired stuff.
Exactly. It also means improving compliance with basic but critical practices, such as meticulous hand hygiene and strict isolation precautions in both hospitals and long-term care facilities. These are fundamental to stopping resistant pathogens from spreading.
So effectively, it's about really tightening up the basics in clinical settings. Makes sense. What's the next step after trying to prevent spread?
The second action is tracking and data. This involves increasing the reporting and sharing of resistance occurrences. Basically, we need a clearer picture of where resistance is emerging and spreading.
Getting ahead of the curve.
That's the goal. This enhanced epidemiological data allows public health officials to identify patterns, pinpoint hot zones, and then target preventive interventions precisely where they're most needed. It's about using real-time information to hopefully outmaneuver the pathogens.
That makes perfect sense. Knowledge is power, right? What about how we're actually using antibiotics? That seems key.
That leads directly to the third core action: antibiotic use and access. A major component here is the requirement for antimicrobial stewardship programs in hospitals.
Stewardship. I've heard that term.
Yeah, it's crucial. These programs track antibiotic usage and educate clinicians on the appropriate and responsible use of these powerful drugs. But it's not just about hospitals. The CDC is also working hard to reduce inappropriate antibiotic use in outpatient settings.
Like your local doctor's office.
Exactly. They've focused heavily on conditions like upper respiratory illnesses, URIs, which are often viral and, don't require antibiotics at all. Overprescribing in these situations is a major driver of resistance. It's about preserving these precious drugs for when they're truly needed.
So using antibiotics wisely is a huge piece of the puzzle. But what about getting new tools in the fight? We can't just rely on the ones we have, especially if they're losing effectiveness, right?
Exactly right. That's the fourth action: vaccines, therapeutics, and diagnostics. Recognizing the urgent need for new weapons, various agencies, including the U.S. Congress and the FDA, have developed incentives and streamlined regulatory pathways.
Trying to encourage development.
Yes. This encourages pharmaceutical companies, both small innovators and large giants, to develop new antimicrobials specifically targeting resistant pathogens. This category also includes the development of rapid diagnostic technologies.
Better tests.
Imagine being able to quickly identify not just the pathogen, but also its specific resistance profile. That allows doctors to choose the right antibiotic much earlier, which is invaluable for effective antimicrobial stewardship.
That would be a game changer, yeah. Moving from a guess-and-check approach to a more targeted one. And finally, the fifth action.
The fifth action is environment and sanitation, which takes a broader, more holistic view. It acknowledges that resistant bacteria and the mobile genetic elements that carry resistance genes don't just exist in human hosts.
Right, they're out there in the world.
They are. They're also found in agriculture, food production, and even domestic animals. This means there's a clearer potential for these resistant strains or genes to be introduced into humans.
Through the food chain, maybe.
Potentially, yes, or direct contact. Therefore, our use of antibiotics in these non-human environments, whether for growth promotion in livestock or disease prevention, can significantly amplify these resistance reservoirs, increasing the risk of human exposure.
Okay.
This is why public health interventions increasingly employ a One Health approach, recognizing that human, animal, and environmental health are fundamentally interconnected. We can't tackle one without considering the others.
That's a truly comprehensive overview of the global battle against microbial threats. It paints a picture of, constant vigilance. With that vital context in mind, let's now zero in on one of those key players mentioned in the urgent category, the pneumococcus.
It's Streptococcus pneumonia.
Our sources take us back in time, revealing its surprisingly long and significant history.
Indeed. The story of Streptococcus pneumonia, or the pneumococcus, begins in the late 19th century. In 1880, quite remarkably, two scientists working independently on different continents made the first observations of this organism.
Pasteur and Sternberg.
That's right, Louis Pasteur in France and George Sternberg, an American army physician. They both recognized these distinct pairs of micrococci when they injected human saliva into rabbits and then examined their blood. What's striking is that, at the time, neither of them immediately grasped the truly devastating role these tiny organisms would play in human disease.
It wasn't obvious at first.
Oh, it took a few more years for that to become clear.
Yeah.
By 1886, the organism was officially named Pneumococcus and Diplococcus pneumonia. By this point, multiple independent investigators had isolated it, and its direct role as a cause of pneumonia was firmly established in the medical community.
And the impact was huge back then.
Immense. Consider this. In the 1930s, pneumonia was the third leading cause of death in the United States, behind only heart disease and cancer. It accounted for about 7% of all deaths in both the U.S. and Europe. And while other pathogens could cause pneumonia, our sources specify that a particular pattern of lung infection, lobar pneumonia, which was overwhelmingly likely caused by the pneumococcus, accounted for roughly half of all pneumonia deaths in the U.S. in 1929 alone.
Half. That's staggering.
It really is. Then, much later, in 1974, a reclassification occurred, and the organism was formally renamed Streptococcus pneumonia, the name we use today.
That's a powerful look back at its origins and impact. So we know its origin story.
Yeah.
But what is this organism from a biological standpoint? What makes it tick, fundamentally?
Okay, from a microbiological perspective, S. pneumonia is a spherical gram-positive bacterium. The term gram-positive refers to how it reacts to a specific laboratory staining procedure, which tells us about the structure of its cell wall.
Okay.
It belongs to the genus Streptococcus, a name derived from Greek roots. Streptos meaning twisted, and coccos meaning berry. This describes its characteristic growth pattern. Its cells divide along a single axis, causing them to form chains or pairs resembling, a twisted string of beads.
Tiny necklaces.
Sort of, yeah. The Streptococcus genus is quite diverse, with at least 22 recognized species, further categorized by their hemolytic properties, basically, how they affect red blood cells when grown in a lab.
And S. pneumonia.
S. pneumonia specifically falls into the alpha hemolytic group. When these bacteria grow on blood agar plates, a special nutrient medium, they produce a distinct greenish color. This green discoloration isn't just random. It's due to a chemical process called alpha hemolysis, where the bacteria reduce the iron within the hemoglobin of the red blood cells. You can actually see this greenish halo around their colonies in lab pictures.
Interesting. What else is characteristic?
In the laboratory, pneumococci are described as fastidious, meaning they have pretty complex nutritional requirements to grow. They're a bit picky. On agar plates, they typically form mucoid, or smooth and shiny, colonies. However, a key insight is that if a pneumococcus lacks its capsule, its colonies will appear rough instead.
Ah, the capsule again.
Exactly. And two crucial features that help identify S. pneumonia and distinguish it from other similar bacteria are its unique sensitivity to a chemical called optochin, which inhibits its growth, and its biosolubility, meaning it dissolves when exposed to bile. These are vital tests for accurate identification in a lab.
So it's got these distinct growth patterns and characteristics that help identify it. But you just mentioned the capsule. From what I understand, that's what truly defines its identity and perhaps its danger.
You've hit on its most defining and arguably most dangerous feature.
Yes.
Like other gram-positive bacteria, S. pneumonia has an inner cell membrane and a strong cell wall. But critically, covering this entire structure is an outer polysaccharide capsule.
A sugary coat.
Essentially, yes. But this isn't just a simple protective layer. Its unique chemical structure is the very basis for classifying pneumococci into different serogroups or serotypes. Historically, scientists distinguished these using what was called the Quellum reaction, where the capsules would visibly swell in the presence of specific antibodies. It was a groundbreaking technique.
So you could actually see the different types under a microscope.
Exactly. It revealed the remarkable diversity of this organism. More recently, with modern scientific advancements, we've discovered even more serotypes, like 6C, 6D, 6F, and many others. These have been identified through sophisticated methods using monoclonal antibodies and advanced genetic and biochemical analyses.
How many are there now?
Our sources tell us there are currently 98 recognized serotypes, grouped into 21 serogroups, each containing 2 to 8 closely related serotypes. And the research continues. Detailed genetic analysis of the specific region of the bacterial genome that codes for this capsule, the Klebsiella, is still revealing new, or putative novel, capsular polysaccharides and variants.
98. That's incredible diversity. So what makes this capsule so critical in terms of causing disease?
This capsule is the primary virulence factor, meaning it's the main weapon that allows the bacterium to cause illness. In the absence of specific antibodies from the host, the capsule acts like an invisible shield.
An invisibility cloak.
That's a good analogy, yes. It prevents the host's immune cells, specifically those involved in phagocytosis, the process where immune cells engulf and destroy pathogens from recognizing and eliminating the bacterium. By evading this initial immune attack, the capsule is arguably the most important determinant of how virulent or harmful the pneumococcus can be. It allows the bacteria to survive, multiply, and cause severe disease.
And it's dynamic, you said.
Very much so. While unencapsulated variants are sometimes found in invasive disease, genetic analysis often shows they do possess the genes for the capsule. This suggests they were likely encapsulated inside the body but start producing the capsule during laboratory isolation. This dynamic expression of the capsule, adapting to its environment, is a key survival strategy.
So it can turn the shield on and off depending on the situation.
Basically, yes. It allows the pneumococcus to evade detection when it's just colonizing, but then raise its shield when it's ready to invade.
It sounds like a highly sophisticated invader with that adaptable shield. Now that we understand what it looks like and how it's classified, let's talk about how this bacterium actually causes disease. What precise tools does it use to invade and thrive within us, beyond just the capsule?
It truly is sophisticated. The pneumococcus possesses a formidable arsenal of molecules located within its cytoplasm, cell membrane, and cell wall, all playing a role in its ability to cause disease or pathogenesis and its virulence. Our sources detail several key players.
Okay, let's break them down.
First, there's pneumolysine, PLY. This is the secreted protein that directly damages and destroys host cells and tissues. It's literally toxic.
A cytotoxin.
Right. Beyond its direct cellular damage, it also powerfully activates our body's complement system, which is a crucial part of our innate immune defense, and stimulates the production of pro-inflammatory cytokines. These are signaling molecules that kickstart and amplify the body's inflammatory response, contributing significantly to the localized damage we see during an infection.
So it damages cells and triggers our own body's damaging response.
Exactly, a double whammy. Of course, the polysaccharide capsule remains paramount. Its primary role is anti-phagocytic, preventing immune cells from engulfing it, mainly by stopping complement proteins from properly binding to the bacterial surface, and it's the main target for protective antibodies.
Okay. What else is in the arsenal?
Then there's pneumococcal surface protein A, PSPA. This protein specifically interferes with the complement system too, blocking a pathway that would otherwise lead to the destruction of the bacteria. It's another clever way the pneumococcus disarms our innate defenses.
2 hits on the complement system already.
At least. PSPC, also known as choline-binding protein A, CBPA, is considered the principal pneumococcal adhesion molecule. Think of it as the bacterium's grappling hook, crucial for sticking to host cells. What's more, it binds to a host protein called factor H, which essentially speeds up the breakdown of C3, another vital component of the complement system, further weakening our defenses.
Grappling hook and shield disabled.
Yeah.
Nasty.
Very. We also have PA and POA pneumococcal iron acquisition A and iron uptake A. These are parts of the bacterial machinery essential for acquiring iron, a critical nutrient for bacterial growth and survival within the host. They're basically iron scavengers.
Gotta get those nutrients.
Right. CBPG, another choline-binding protein, plays a role in cleaving or cutting the host's extracellular matrix. This matrix is the scaffolding that holds our tissues together. By degrading it, CBPG helps the bacteria adhere better and likely facilitates their spread through our tissues.
Taking down the walls.
Precisely. PSA, or pneumococcal surface antigen A, is a metal-binding protein specifically for zinc and manganese. It's also thought to contribute to adhesion, another anchor point for the bacterium. The pneumococcus also produces hyaluronate lyasis, an enzyme that breaks down hyaluronan and chondroitin sulfate, key components of that extracellular matrix, again helping the bacteria invade and spread.
More matrix destruction.
Then there's IgA1 protease. This enzyme specifically degrades human IgA1, a type of antibody found on our mucosal surfaces, like in the nose and throat. By chopping up this antibody, the pneumococcus effectively evades a key part of our immune response right at the entry points.
Taking out the guards at the gate.
An intriguing one is autolysin, LyTA. This enzyme causes the bacterial cell to self-destruct, or autolyse. When it does, it releases various intracellular contents, including parts of the cell wall, teichoic acid, and pneumolysin itself. This release of bacterial components triggers a strong inflammatory response from the host.
Wait, it kills itself to cause more damage.
It seems counterintuitive, but yes. That burst of inflammatory material contributes to the disease symptoms. The bacterium also uses neuraminidase, NANB. These enzymes help the pneumococcus adhere to host cells by removing specific sugar molecules called sialic acids from host glycoproteins and mucin. This removal exposes new binding sites that the pneumococcus can then attach to more effectively.
Clearing the way for attachment.
Exactly. Additionally, pili are present on the bacterial cell surface. These are tiny, hair-like appendages that not only help the bacteria stick to cells, but also inhibit phagocytosis and promote invasion. And finally, a very important group are the penicillin-binding proteins, PBPs.
Ah, the PBPs. Crucial for resistance later.
Absolutely crucial. These are enzymes vital for building the bacterial cell wall. They connect the building blocks. Understanding PBPs is absolutely critical for comprehending how antibiotic resistance works, especially to penicillin, which targets these very proteins to disrupt cell wall construction.
Wow, that's an incredibly sophisticated array of tools working at multiple levels to evade our defenses and cause damage. So how does this elaborate set of virulence factors translate into the actual process of infection once the pneumococcus gets into the body? How does this biological machinery play out in a human host?
It's a journey, really, from initial contact to potentially severe disease. The first and most crucial step is nasopharyngeal colonization. Pneumococci frequently colonize the human nasopharynx, that's the area behind your nose and above your throat.
Just hanging out there.
Often, yes. Starting from a very early age, sometimes even days after birth in high-burden regions. While these colonization events are often asymptomatic, meaning they don't cause obvious illness, recent research suggests that acquiring a new strain can sometimes be linked to mild respiratory symptoms, especially in very young children. Once in the nasopharynx, these bacteria are remarkably adept at survival. They're protected by their capsule, and they form a biofilm.
Like a bacterial city.
A sticky community, yeah, encased in a self-produced matrix. Within this biofilm and the mucus secreted by our epithelial cells, they're shielded from local immune factors like white blood cells and complement. Our body also has that natural defense mechanism, the mucociliary escalator.
The cleaning crew.
Right, constantly sweeping bacteria out.
Yeah.
The pneumococcus has to contend with this mechanical clearance too. While many colonization episodes are brief, studies have shown that persistent colonization with a specific serotype can last for many months.
Months. Just living in your nose.
It can happen. From the nasopharynx, the bacteria can spread in two main ways. The first is hematogenous spread. They enter the bloodstream.
Travel system-wide?
Exactly. Once in the blood, they can travel to distant sites like the brain, joints, bones, or the abdominal lining, leading to severe systemic infections. The second pathway is local spread, where they move directly to adjacent mucosal surfaces. This causes conditions like otitis media, ear infection, or pneumonia.
So bloodstream or local invasion.
Right. It's worth clarifying that direct spread from the nasopharynx to the brain, causing meningitis, is rare. Usually only happens with something like a skull base fracture. Most cases of pneumococcal meningitis actually follow that hematogenous spread.
That's a key distinction. So the nose and throat are really the starting point for everything. And you mentioned the capsule's dynamic nature earlier. Did that play a role here too?
Absolutely, and it's a brilliant adaptive strategy. The pneumococcus dynamically modifies its capsule expression in response to its environment. For instance, when it's just colonizing the nasopharynx, it tends to downregulate capsular expression.
Keeping a low profile.
Exactly. Makes it less visible to our protective immune mechanisms that would otherwise target the capsule. When cultured from the nasopharynx in this state, they produce those rough colonies. However, when the bacterium invades the body, crossing the epithelial barrier and beginning to cause disease, it quickly upregulates its capsular expression.
Shields up.
Precisely. This transformation makes its appearance in culture change to smooth colonies and is critical for it to evade immune detection and cause invasive disease. It's remarkable how effective our immune system is, though, because even though colonization is frequent, actual disease is relatively rare.
Thank goodness.
When colonization occurs, our body eventually develops specific IgG antibodies, a type of protective protein that target both the capsule and surface proteins. These antibodies are thought to be key in clearing the bacteria from the nasopharynx.
So we learn to fight it off eventually.
Yes, But a critical factor that makes someone vulnerable to invasive disease is the recent acquisition of a new colonizing serotype. This is more often linked to subsequent invasive disease, likely because the host hasn't yet developed specific immunity to that particular new strain.
Ah, the element of surprise.
In a way, another crucial piece of the pathogenesis puzzle is the strong association between intercurrent viral infections like the flu or a common cold and an increased susceptibility to pneumococcal colonization and disease.
The old secondary bacterial infection after a cold.
That's exactly it. Often, pneumococcal disease in someone already carrying the bacterium follows a disruption of the nasopharyngeal mucosa by a viral infection. These viral infections trigger local cytokine production, which in turn is thought to boost the expression of adhesion factors on our respiratory epithelial cells. This allows pneumococci to adhere much more effectively using their various surface adhesin molecules, PSIA, PSPA, CBPA, PSPC, high-hyl, pneumolysin, neuraminiditases, all those tools we discussed.
So a viral infection basically lays out the welcome mat for the pneumococcus, making it easier to attach and then invade.
Precisely. And this enhanced adhesion, or combined with the inflammation caused by bacterial components like cell wall-derived teichoic acids and peptidoglycans, then kicks off an intense inflammatory response. These bacterial components trigger our body to produce powerful pro-inflammatory cytokines such as IL-1, IL-6, and TNF.
The inflammation cascade.
Exactly. This cascade also activates our complement system. All of this attracts polymorphonuclear leukocytes, a type of white blood cell, to the site of infection. Pneumococcal also directly contributes to this local inflammation by inducing cytokine production from our monocytes. While many capsular types can cause human disease, certain types disproportionately cause invasive pneumococcal disease, though the exact reasons for this dominance aren't fully understood.
It's clear that the pneumococcus is a formidable opponent with all those tools and adaptive strategies, but our bodies have incredible defense systems. How do we naturally fight back against these invaders, and what are the key components of our immune response? Getting into the host defense mechanisms.
That's the other side of this intricate battle, yeah. And our body's defense mechanisms against pneumococci are multi-layered, involving both innate immunity, our immediate non-specific defenses, and acquired immunity, which learns and adapts.
Let's start with innate, the first responders.
Right. Our innate immunity is our body's first line of defense, always ready to act. This includes crucial physical barriers like the intact respiratory epithelium, the protective layer of mucus in our airways, our cough reflex, and the mucociliary escalator we mentioned, constantly sweeping bacteria out.
Physical removal.
Exactly. Beyond these physical barriers, key nonspecific immune factors are vital. Proper splenic function, the complement system, neutrophils, and macrophages. These are all types of immune cells and proteins that work together to identify and clear bacteria.
Okay. And specific immune molecules.
Digging deeper into the immunologic factors, C-reactive protein, CRP, plays a significant role. It's a protein that binds to a specific component in the pneumococcal cell wall called phosphorylcholine. This binding then triggers complement activation, leading to bacterial clearance.
So CRP flags it for destruction.
Essentially, yes.
Huh.
Then we have the toll-like receptors, or TLRs, which are essential for recognizing pathogens. For example, TLR2 recognizes certain pneumococcal components, and studies show that if this receptor is absent, infections become more severe and bacteria aren't cleared as effectively from the nasopharynx. Similarly, TLR4 is necessary for the powerful inflammatory effect that pneumolysin has on macrophages.
So these TLRs are like pattern detectors.
Precisely. The absolute importance of TLR recognition is profoundly underlined by genetic conditions like the inherited deficiency of human IL-1 receptor-associated kinase 4, IRP4. Individuals with this deficiency are unusually susceptible to bacterial infections, including S. pneumonia, because IRC4 is essential for the normal function of several TLRs.
So it's like a critical missing piece in their early warning system. And I imagine that if any of these innate defenses are compromised, it opens the door for infection.
Exactly. Factors that interfere with these non-specific mechanisms significantly predispose individuals to pneumococcal pneumonia. Our sources list several, including viral infections, cystic fibrosis, bronchiectasis, complement deficiencies, and COPD. Another critically important predisposing factor involves splenic function.
The spleen. Why is that so important?
Patients who lack a spleen or have abnormal splenic function, such as individuals with sickle cell disease, are at an extremely high risk of developing overwhelming pneumococcal disease. The spleen is a vital organ for filtering bacteria from the blood and mounting an effective early immune response, especially against encapsulated bacteria like pneumococcus.
Okay, that makes sense. Now, shifting to acquired immunity, the
Yeah.
learning part of the system.
Right. This is how our body learns to fight back more effectively the next time it encounters a pathogen. This immunity, whether developed after a natural colonization episode or through exposure to other cross-reactive antigens, largely relies on the development of specific IgG antibodies in the blood that target the pneumococcal capsular polysaccharide.
So antibodies against that sugary coat.
Exactly. That's the primary target for acquired immunity.
That makes sense.
Hmm.
Our body builds up a memory, so to speak. But is it the same for everyone, or are there differences, say, in children?
That's a key point. What's interesting about most polysaccharides, including the pneumococcal capsule, is that they are considered T-cell independent antigens. This means that our B cells, a type of immune cell, can produce antibodies to these antigens without direct help from T cells.
Okay.
However, in children less than 1 to 2 years old, these B cell responses are not fully developed. This delayed development or ontogeny of capsule-specific IgG in very young children directly explains their increased susceptibility to pneumococcal infection. It's a critical period of vulnerability in pediatric care.
Their immune systems are still learning.
Right. The absolute importance of capsular antibody for protection against disease is further highlighted by the extremely high risk of pneumococcal infection in individuals who lack sufficient serum immunoglobulin, as seen in conditions like agemoglobulinemia.
Underscores the antibody role and the different serotypes. Does immunity to one protect against others?
Generally, no. Because each serotype's capsule is chemically distinct, the immunity we develop tends to be serotype specific. However, there are instances of cross-immunity. For example, antibodies induced by conjugate vaccines against serotype 6b can also help prevent infection from serotype 6a.
Some overlap, then.
Some, but it's important to note that cross-protection between serotypes within the same serogroup isn't universal. For instance, antibodies to serotype 19F from some vaccines don't appear to protect against disease caused by serotype 19A. It's quite specific.
Tricky. Are there other targets besides the capsule for acquired immunity?
Yes. Beyond capsular antibodies, our body also produces antibodies to other surface-exposed or secreted pneumococcal proteins, such as pneumolysin, PSA, and PSPA. These protein-specific antibodies also appear in circulation as we age or after colonization, and they likely contribute to overall protection against disease.
So a broader immune response develops over time.
It seems so. Finally, data from animal models, like studies in mice, suggest that CD4 plus T cells, another type of immune cell, may also play a role in preventing pneumococcal colonization and disease, and human experimental data also support this idea, pointing to a more complex interplay of immune responses at work than just antibodies alone.
It's truly a complex dance between the invader and our defenses. But when the pneumococcus does manage to break through, what does that actually look like for a patient? How do doctors recognize and deal with it, given its ability to manifest in so many different ways?
You've hit on one of the biggest challenges in clinical practice. There is no single telltale presentation for pneumococcal disease. Patients can show up with one or more clinical syndromes: pneumonia, meningitis, sepsis. Pneumonia has the capacity to infect nearly any body tissue.
The kit's everywhere.
It can, and the severity can range wildly, from a mild, self-limited illness to a life-threatening emergency. Because of this variability, pneumococcal infection must always be considered in the differential diagnosis for common syndromes like pneumonia, otitis media, fevers of unknown origin, and meningitis.
Always keep it on the list.
Always. A critical point is that a microbiologically confirmed diagnosis is actually made in only a minority of pneumococcal cases.
Why is that?
Because, especially with pneumonia and otitis media, it's often difficult to get samples directly from the infection site to identify the bug. And even when someone has pneumonia, the bacteria only show up in the bloodstream in a minority of cases, less than 30%.
So you often don't have definitive proof.
Exactly. So often, doctors have to start empirical therapy treatment based on the most likely cause before a definitive diagnosis that includes appropriate coverage for S. pneumonia. In settings with limited resources, standardized algorithms like the Integrated Management of Childhood Illness, IMCI, are used for kids. These guide healthcare workers to identify danger signs and start antibiotics quickly.
But not for adults.
No similar standardized algorithms exist for adults, which adds to the complexity. Clinically, we classify syndromes broadly into non-invasive illness, like otitis media, where it spreads locally, and invasive disease like bacteremic pneumonia or meningitis, where it's in normally sterile sites. But remember, all pneumococcal infections fundamentally originate from that nasopharyngeal acquisition.
Back to the nose and throat.
Always. Let's do a deep dive into pneumonia, as it's the most common serious pneumococcal syndrome. There's even debate about whether non-bacteremic pneumococcal pneumonia should be classified as invasive or not invasive. Its presentation can range from mild community-acquired to life-threatening.
What are the typical symptoms, then, for pneumonia caused by this bacterium? Is there anything that makes it stand out?
Unfortunately, its presentation usually doesn't reliably distinguish it from pneumonia caused by other pathogens. It's often preceded by a viral URI. Patients commonly experience an abrupt onset of cough and dyspnea, shortness of breath, accompanied by fever, shaking chills, and muscle aches.
Sounds like a bad flu almost.
It can feel like that initially. The cough often starts non-purulent and then progresses to producing purulent, sometimes even blood-tinged sputum. Patients may also describe a sharp, stabbing pleuritic chest pain, indicating inflammation of the lung lining. This pain can make breathing difficult.
Ouch. What about older patients? You mentioned they could be different.
Yes, and this is crucial. Elderly patients might have far less specific symptoms. They might present with confusion or general malaise without the classic fever or cough. In these situations, a very high index of suspicion is required because delaying treatment can lead to rapid deterioration.
Got to be vigilant with the elderly. What signs might a doctor find on exam?
On physical examination, adults often show tachypnea, fast breathing, and tachycardia, fast heart rate. Severe cases might have hypotension, low blood pressure. Most patients have a fever, but again, not always the elderly. Lung sounds can include dullness to percussion over areas of consolidation and crackles. Less commonly, bronchial breathing or a pleural rub. Cyanosis, that bluish skin color, signals very severe disease.
And infants?
In infants with severe pneumonia, characteristic findings include chest wall indrawing and nasal flaring. Non-respiratory findings can also occur, like upper abdominal pain if the diaphragm is involved, and mental status changes, especially confusion in older adults.
That's a wide range of symptoms, making diagnosis tricky. What else would a doctor consider when someone comes in with these symptoms? What's the differential?
The differential diagnosis is broad. It includes cardiac conditions like a heart attack or heart failure, other lung conditions like atelectasis, and pneumonia caused by other pathogens, viruses, mycoplasmas, other bacteria like H. flu, Klebsiella, Staph aureus, Legionella, or in immunocompromised hosts, pneumocystis. And if abdominal symptoms are prominent, you have to think about abdominal issues like cholecystitis or appendicitis, but also remember that pneumonia can cause referred pain.
A lot to rule out. How's the diagnosis usually made then?
For diagnosis, some experts suggest treating uncomplicated, non-severe community-acquired pneumonia empirically. But for more severe or unclear cases, identifying the cause is crucial. The gold standard is examining lung tissue, but that's rarely done.
Right, too invasive usually.
Exactly. In practice, evidence of an infiltrate on chest radiography generally confirms pneumonia. Classically, pneumococcal pneumonia shows lobar or segmental consolidation, though it can be patchy. Sometimes involves more than one lobe. In children, round pneumonia suggests pneumococcus, but that's rare in adults and needs careful evaluation.
So imaging is key. What about blood tests or other lab work?
Blood tests help. Blood cultures are positive for pneumococci in less than 30% of cases. Nonspecific findings include a high white blood cell count, though sometimes it can be low, leukopenia, which is a bad sign. You might also see elevated liver enzymes, anemia, low albumin, low sodium, or kidney issues.
What about those urine tests?
Urinary pneumococcal antigen assays exist. They detect a cell wall component, but interpreting them is tricky. Simple nasopharyngeal carriage can cause a positive test. So in adults where carriage is less common, a positive test is more suggestive of disease. But in kids with high carriage rates, it's usually uninformative.
So useful in adults, less so in kids.
Generally, yes. Newer quantitative serotype-specific urine tests show promise for both. In many adult cases, diagnosis still relies on gram staining and culture of sputum. But the quality of the sputum sample and whether antibiotics have already been started really affect the results.
Beyond pneumonia, what are the most common and concerning complications and other serious syndromes it causes?
When it comes to complications of pneumococcal pneumonia, empyema is the most common focal one, occurring in less than 5% of cases. That's pus collecting in the pleural space.
Sounds nasty.
It is. You suspect it if there's fluid plus persistent fever and high white count after several days of antibiotics. Parent pneumonic effusions, just inflammatory fluid, are more common and often resolve. But true MPMA, identified by pus or specific fluid characteristics like low pH, requires aggressive drainage, usually with a chest tube.
Okay. And meningitis.
Pneumococcal meningitis typically presents as pyogenic meningitis, clinically indistinguishable from other bacterial types. It can be the primary syndrome or complicate other things like skull fracture or ear infection. Since the Hib vaccine reduced H. influenza, S. pneumonia, and Neisseria meningitis are now the most common bacterial causes in adults and kids.
What are the meningitis symptoms?
Severe generalized headache, often gradual onset, fever, nausea, specific CNS signs. Stiff neck, photophobia, seizures, confusion. Patients might look toxic, have altered consciousness, slow heart rate, high blood pressure from intracranial pressure. Classic signs like Kernig's or Brudzinski's are only present in a minority.
So how is meningitis diagnosed? And what are the outcomes? It sounds very serious.
Diagnosis is confirmed by examining the cerebrospinal fluid, CSF, via lumbar puncture. Look for turbidity, high protein white cells, low glucose. Then identify the bug with culture, gram stain, antigen tests, or PCR. A positive blood culture with clinical meningitis also confirms it. The urine antigen test is highly specific in adults again.
And the prognosis?
Mortality is around 20%, and up to half of survivors have complications. In kids, deafness, hydrocephalus, mental retardation. In adults, brain swelling, bleeding, hydrocephalus, stroke-like issues, hearing loss. It can be devastating.
Truly devastating. What other invasive forms are there?
Other invasive syndromes include primary bacteremia without another focus, occult bacteremia, bone infections, osteomyelitis, joint infections, septic arthritis, heart valve infections, endocarditis, pericarditis, peritonitis. Diagnosis involves getting sterile fluid from the site for gram stain, culture, maybe antigen or PCR. And a rare but serious complication of invasive disease is hemolytic uremic syndrome.
Okay. And briefly, the non-invasive ones.
The main non-invasive syndromes are sinusitis, bacterial bronchitis, and acute otitis media, AOM. AOM is the most common pneumococcal syndrome overall, especially in young kids.
The classic ear infection.
Exactly. Acute onset of severe ear pain, fever, sometimes deafness or tinnitus, often after a cold. The eardrum looks red, swollen, often bulging. Pneumococcal sinusitis also often follows a cold, causing facial pain, congestion, fever, persistent cough. Diagnosis is often clinical, though sinus aspiration is definitive. Pneumococcal bronchitis usually hits people with existing lung conditions like COPD. Interestingly, this might be caused by non-typable strains, those without a capsule.
That's an incredible clinical overview, truly highlighting the diverse ways this bacterium can attack. Now that we understand how it causes disease and how it manifests, let's shift our focus to its broader movement. How does it spread through populations? And how have our interventions, especially vaccines, fundamentally changed its landscape? It sounds like a continuously shifting battlefield.
It absolutely is a dynamic landscape, and the epidemiology reveals profound global health implications. Pneumococcal infections remain a significant global cause of illness and death, disproportionately affecting children and the elderly.
The numbers are stark, aren't they?
They are. Our sources estimate around 317,000 deaths worldwide among children aged 1 to 59 months back in 2015. That's nearly 10% of all child deaths and almost 40% of all pneumonia deaths in that age group. Getting reliable global estimates for adults is harder due to limited data, especially from high-burden regions.
Where are the highest rates?
The highest rates are concentrated in selected countries of sub-Saharan Africa and Southern Asia. This disparity is often linked to prevalent risk factors: HIV infection, lack of breastfeeding, malnutrition, sickle cell disease, and crucially, limited access to medical care.
So it's very much a global health equity issue. What about the different types of pneumococcus? Do certain serotypes cause more problems than others?
That brings us to serotype dynamics. Yes, not all pneumococcal serotypes are equally likely to cause disease. Their distributions vary significantly by age, the specific disease syndrome, and geography. Most available data on serotype distribution come from pediatric invasive pneumococcal disease, IPD infection, of a normally sterile site. Less info for adults globally.
What did it look like before vaccines?
In the pre-PCV era, before widespread conjugate vaccine use, a relatively small number of serotypes, usually 5 to 7, caused over 60% of IPD cases in young children globally. Specifically, 7 serotypes, 1, 5, 6A, 6B, 14, 19F, and 23F, accounted for about 60% of these cases worldwide, though their rank varied.
A handful causing most of the trouble.
Pretty much. Some serotypes, like 1 in 5, have unique behaviors, causing waves of disease even in lower burden areas or specific outbreaks. But the widespread use of pneumococcal conjugate vaccines has profoundly altered this epidemiology.
How so?
Now, serotypes that weren't prominent before are emerging as important causes of invasive disease, things like 15BC, 22F, 10A, 23B, and others. It's also noteworthy that some serotypes included in the vaccines, like 3 and 19A, continue to cause IPD in many settings. Persistent challenges remain.
That's a powerful example of how public health interventions can literally reshape the microbial landscape. What about how these bacteria spread from person to person, the carriage aspect?
That leads to nasopharyngeal carriage, which is key to transmission. Pneumococci are intermittent inhabitants of the healthy human nasopharynx, spread mainly by respiratory droplets.
So living in our noses again.
Yep. In children, carriage patterns vary a lot based on region, socioeconomic status, climate, crowding, and especially exposure to other kids. Daycare is a big factor.
Makes sense.
In developed countries, kids are the major vectors. About half of children have had at least one colonization episode by age 1. Prevalence rates might be 20–50% in kids under 5, but only 5–15% in young and middle-aged adults.
What about lower-income countries?
In low and middle-income countries, LMICs, acquisition happens much earlier, sometimes within days of birth. Nearly all infants are colonized by 2 months. Studies show 70–90% of kids under 5 carry it, and often over 40% of adults too.
Much higher adult carriage there.
Exactly. Which makes adults important sources of transmission too, affecting community dynamics differently.
That's quite a difference. So what has the impact of PCV been on actual disease rates? Given these shifts in serotypes and carriage, I imagine it's not a simple story of universal decline.
You're right, it's nuanced, especially with serotype replacement. Remember, IPD significantly underestimates the true burden of pneumonia because most pneumonia isn't bacteremic.
Right, only 10–30% show up in blood cultures.
Exactly. Pre-PCV, U.S. data showed highest IPD rates in kids under 2 and adults over 65. Since PCV introduction, IPD rates in U.S. infant children have plummeted by over 75%, driven by near elimination of vaccine serotype IPD. Similar impacts are seen wherever pediatric PCV is routine.
That's huge success. But what about the non-vaccine types?
That's where it gets complex. Changes in non-vaccine serotype IPD rates have been heterogeneous. In the U.S., Canada, Australia, these rates increased, but the increase was generally small compared to the massive vaccine serotype productions.
So overall, still a big win.
In those places, yes, but in others, like Alaska Native communities and adults in the UK, reductions in vaccine serotype IPD were significantly offset by notable increases in non-vaccine serotypes. This has eroded the original vaccine impact there.
So it's almost like the vaccine creates a vacuum and other strains step in to fill the niche. What factors contribute to this varying degree of replacement?
That's a good way to put it. The explanations are varied: direct replacement disease from vaccine pressure, changes in clinical investigation, underlying secular trends unrelated to vaccines, antibiotic pressure, changes in surveillance, how quickly PCV was rolled out, use of catch-up campaigns. It's likely a mix of factors.
Complex.
Very. But the key takeaway is that while serotype replacement does occur post-PCV, its magnitude is generally small compared to the vaccine serotype disease reduction in vaccinated populations, except, again, in places like the UK for adults, where non-vaccine types are really eroding the impact, and types 3 and 19A persist despite being in the vaccine.
And pneumonia itself remains a challenge diagnostically.
Yes, because most is non-bacteremic. PCV trial showed only 5–20% of pneumonia cases are bacteremic, and the Capita trial confirmed similar findings in elderly adults. Diagnosis relies on good sputum or, in adults, urine antigen tests. Plus, there's growing evidence for co-infection.
Right, pneumonia often isn't a single bug fight.
Exactly. It's often sequential or contemporaneous co-infection, maybe virus plus bacteria. Single pathogen pneumonia might actually be uncommon.
That's a crucial insight. What about the fatality rates? How do they vary, and what influences them?
Case fatality ratios, CFRs, vary a lot by age, underlying conditions, and access to care. For pneumonia, CFR changes with severity and site of care, not just whether it's bacteremic.
Interesting.
A surprising observation from our sources is that the likelihood of death in the first 24 hours of hospitalization didn't substantially change with the introduction of antibiotics.
Antibiotics didn't impact early deaths.
Not substantially, according to the data cited. It highlights that severe pneumococcal pneumonia involves a rapid, progressive cascade, often independent of initial antibiotics. ICU management provides critical support, leading to lower CFRs, while antibiotics treat the underlying infection over time.
That's quite a sobering thought. Antibiotics alone aren't always enough to stop that rapid early deterioration. Beyond geography and age, what other factors increase someone's risk of getting these infections?
Rates are higher in colder months in temperate climates. Males more affected than females. Risk factors include underlying medical conditions, behaviors like smoking, and certain ethnic groups. In the U.S., some Native American populations and African Americans have higher rates, likely due to socioeconomic factors and underlying risk prevalence.
What specific medical conditions?
Our sources list quite a few: asplenia or splenic dysfunction, like sickle cell, chronic respiratory diseases, COPD, asthma, chronic heart, kidney, or liver disease, diabetes. Also, immunocompromise from HIV, cancer, chemotherapy, transplants, long-term steroids, plus cochlear implants or CSF leaks.
A long list. Any others?
Miscellaneous risks include infancy and old age, prior hospitalization, alcoholism, malnutrition, smoking, daycare attendance, residence in crowded settings like military camps or shelters. Outbreaks happen in these places. And again, that clear link with preceding viral infections, especially flu. Secondary pneumococcal infections are a major cause of illness and death during flu season and pandemics.
So we've talked about its epidemiology and how it moves through populations, as well as the risk factors. But despite our best efforts, it keeps finding ways to survive. Let's delve deeper into one of the biggest ongoing threats it poses: antibiotic resistance. How did this problem emerge, and what makes it so challenging?
This is truly a critical area. Reduced pneumococcal susceptibility to penicillin was first noted way back in 1967.
That far back.
Yes, but it wasn't until the 1990s that reduced antibiotic susceptibility really emerged as a significant clinical and public health issue. That's when we saw an increasing prevalence of isolates resistant to single or multiple antibiotic classes and a rising absolute number of resistant strains.
So it accelerated in the 90s.
It did. Today, reduced susceptibility to key antibiotics like penicillin G, cefotaxime, ceftriaxone, and macrolides is found worldwide. These resistant strains account for a significant proportion of disease, especially in kids.
Any good news on the resistance front?
One crucial point of relative relief is that vancomycin resistance has not yet been observed in clinical pneumococcal strains.
That's huge.
Yeah.
Vandic is often the last resort.
It is. But it's also important to understand that lack of antimicrobial susceptibility is clearly related to a subset of specific serotypes, many of which disproportionately cause disease among children.
So resistance travels with certain strains. How exactly does pneumococcus become resistant? What are its tricks?
The mechanisms of resistance are complex. It involves a mix of mutational events in their own DNA and intraspecies gene transfer.
So they can swap genes with other bacteria.
Exactly, often using mobile genetic elements like plasmids or transposons. This horizontal gene transfer allows successful resistant clones to spread rapidly. Specifically for penicillin resistance, the main mechanism is altering the penicillin-binding protein, PBP genes.
Those crucial cell wall.
Right. They get altered through transformation, picking up DNA, and horizontal transfer from related species. These altered PBPs have a lower affinity for penicillins, so the drug can't bind as well. The level of resistance depends on which PBPs are altered and how many. It's quite sophisticated.
So it changes the very target that the antibiotics are trying to hit. And this, I imagine, creates a vicious cycle.
It absolutely does, the classic vicious cycle. Antibiotic exposure selects for resistant organisms in the nose. They spread, cause hard-to-treat infections, which leads to more antibiotic use, driving more resistance.
The feedback loop.
Exactly. To some extent, the introduction of PCV has interrupted this cycle, which is a major public health win. But despite that, multi-drug-resistant pneumococci are still considered a serious threat by the CDC.
So if we have these resistant strains, how does that change how we treat these infections? What are the current guidelines for medical professionals trying to combat these evolving bacteria, especially in the context of increasing resistance?
Resistance has indeed fundamentally altered treatment. Historically, parenteral penicillin G was the go-to for susceptible bugs. Today, parenteral beta-lactam drugs like ampicillin, cefotaxime, ceftriaxone, cefuroxime are often first line for community-acquired infections.
And for penicillin allergies.
Macrolides and cephalosporins are alternatives. Agents like clindamycin, tetracycline, trimethoprim, sulfamethoxazole have some activity, but resistance is common worldwide, making them less reliable empirically.
So how do doctors know if a strain is resistant? Those breakpoints.
Yes, the resistance breakpoints are key. Penicillin resistance emerged mid-60s, multi-drug resistance in the 70s, becoming pandemic in the 90s. The Clinical and Laboratory Standards Institute updated penicillin G breakpoints in 2008 because the old ones didn't always predict patient response well, except for meningitis.
Okay, what are the current breakpoints?
For IV treatment of meningitis with high-dose penicillin, susceptible is ALE 0.06 ecml, resistant is EO 0.12 ecml, resistant is ERA 0.1 FANNL. For AV treatment of non-meningeal infections with standard doses, susceptible is A2 ecml, resistant is A8 ecml. Intermediate strains need higher doses.
So those specific numbers are crucial for guiding doctors. What are the general guidelines for treatment, and how do they differ for specific conditions like meningitis? That seems like the most critical scenario.
General therapy guidelines should partly consider local resistance, but national organizations provide evidence-based approaches. For kids, the AAP Red Book is key. Let's look at specific syndromes. For meningitis likely or proven to be S. pneumonia. In high-resistance areas or after travel, first line for anyone over a month old is a combination of vancomycin plus a third-gen cephalosporin like cefotaxime or ceftriaxone.
Covering all bases.
Right. But in low-prevalence areas without travel history, vancomycin is not included first line. For kids with severe beta-lactam allergies, yfempin can substitute or be added.
And what if the patient isn't improving or if the resistance profile is still unknown? Do you recheck the spinal fluid?
Yes, a repeat lumbar puncture after 48 hours should be considered in those situations, or if dexamethasone was given, as it can mask response. Once sensitivity data arrive, you tailor treatment.
How so?
If it's penicillin sensitive, stop vanc. Penicillin can replace the cephalosporin or continue the cephalosporin alone. If penicillin resistant but cephalosporin susceptible, stop vanc, continue the cephalosporin. If resistant to both penicillin and non-susceptible to the cephalosporin, continue vanc and high-dose cephalosporin. Maybe add rifampin.
What about steroids? I hear they're used in meningitis.
Corticosteroids show benefit in high-income countries. They reduce mortality, hearing loss, neurologic problems in adults if given early. But they don't seem beneficial in low-income countries, possibly due to delays in care.
That's a critical nuance depending on the setting. What about sepsis beyond meningitis?
For sepsis, excluding meningitis, in non-critical kids, standard antibiotics like ampicillin, cefotaxime, ceftriaxone, or penicillin G work. For critically ill kids, myocarditis, severe pneumonia, vancomycin might be added if resistance is suspected, pending susceptibility results. If resistant, therapy is guided by susceptibility testing. For severe beta-lactam allergy, clindamycin or vancomycin are options, but stop vanc if sensitive to other non-beta-lactams.
And for pneumonia that can be managed outside of the hospital, what's the go-to?
Outpatient pneumonia. Oral amoxicillin, usually high dose, is effective for almost all cases.
Still good old amoxicillin.
Yep. Cephalosporins offer no real advantage and cost more. Fluoroquinolones like Levo or Moxie are usually effective in the U.S. unless there's prior exposure risk. Clindamycin works well. Macrolides like azithromycin work. But a critical warning. Treatment failure leading to bacteremic disease due to macrolide-resistant isolates has been well documented with empirical azithromycin. So, while resistance varies, high-dose amoxicillin remains the best global option for outpatient pneumonia.
So even with all these advanced options, simple high-dose amoxicillin is still often the best choice globally. What about the duration of treatment for pneumonia? How long?
The optimal duration is actually uncertain, but treating for at least 5 days after the fever breaks seems prudent. In adults, 5 days total often suffices. Longer therapy is needed if there's another infection site, like empyema.
Okay, and ear infections, AOM.
For acute otitis media, AOM, high-dose amoxicillin is recommended for infants 6 months, and those 6-23 months with bilateral disease. For older kids with non-severe illness, observation might be an option. Duration is usually 10 days for younger kids or severe disease, maybe 5-7 days for older kids with mild-moderate illness.
What if it doesn't get better?
Reassess in 48-72 hours. If AOM confirmed, start amoxicillin if not already on it. If initial therapy fails, change antibiotics. If second line, like amoxicillin clavulinate, fails, may need myonotomy or tympanocentesis to get fluid for culture.
Get a sample to know what you're fighting.
Exactly. Sinusitis recommendations often follow AOM guidelines. Many professional groups have published detailed guidelines for kids.
That's an incredibly detailed, truly professional level understanding of how these infections are tackled. But prevention is always better than cure, right? Especially with the persistent threat of resistance. What can we do to stop these infections before they even start?
Absolutely right. Prevention is paramount, and the strategies are multifaceted, focusing heavily on vaccination. General measures include, first, vaccination against S. pneumonia and influenza, also, reducing underlying comorbidities, and crucially, preventing antibiotic overuse.
Let's talk vaccines. Start with the older one, PPSV23.
Right. The capsular polysaccharide vaccine, PPSV23, it covers 23 serotypes, licensed since '83. In the U.S., ACIP recommends it for everyone 065 and for those 264 with underlying conditions increasing risk. Updated recommendations often involve using a conjugate vaccine first, then PPSV23.
So it's not a one-and-done vaccine for everyone. What about booster shots?
Revaccination is recommended 5 years after the first dose for those over 2 with underlying conditions, but not routinely just for being 065. Why? PPSV23 doesn't give a strong memory response. Antibodies wane, so boosters are key for those prone to antibody loss.
Are there downsides to boosters?
Concerns exist about repeated revaccination local reactions and potential immune hyporesponsiveness, where the immune system responds less well. The clinical impact isn't totally clear, but because of it, more than one revaccination hasn't generally been recommended.
And how well does PPSV23 actually work? Looks like there's debate.
Its effectiveness is controversial. Observations vary wildly for preventing IPD, pneumonia, death. Meta-analyses disagree. Observational studies usually show more benefit than controlled trials.
What's the general consensus?
Generally effective against IPD, but less so against non-bacteremic pneumonia, though results conflict. Effectiveness is often lower in the elderly and immunocompromised. Protection lasts maybe 5 years. What's not disputed is the need for better adult vaccines, as non-conjugate types still cause significant disease despite infant PCV use.
So there's still a significant gap in adult protection, which brings us to the conjugate vaccines. How do they differ, and why were they developed? PCVs.
Exactly, polysaccharide protein conjugate vaccines, PCVs. The rationale was simple. Infants and young kids respond poorly to PPSV, those T-cell independent antigens. PCVs were specifically designed for them by linking the polysaccharide to a protein carrier, which does engage T-cells.
Making it work better for young immune systems.
Precisely. The first was 7-valent, PCV7 in 2000. Now, 10 and 13-valent versions, PCV10, PCV13, are widely available. They target key serotypes causing IPD and resistance in kids.
And the evidence.
Randomized controlled trials proved high efficacy against vaccine serotype IPD, plus efficacy against pneumonia, otitis media, colonization, even all-cause mortality. Huge success.
That's why they're recommended globally now.
Yes, WHO recommends them worldwide, especially in high infant mortality countries. As of our sources, 144 countries use them, 16 planning to, 34 haven't decided.
That's a truly global adoption and a massive public health achievement. What kind of impact has this had in countries that widely adopted them?
Dramatic impact in high-income settings. Over 90% reduction in vaccine serotype IPD across the whole population. A key concept is indirect effect, or herd immunity.
Protecting the unvaccinated, too.
Right. Reduced colonization in vaccinated kids means less spread to adults, but we also see replacement disease increases in non-vaccine types. The scale varies, significantly eroding impact in UK elderly, less so in the USA.
So a trade-off sometimes. Any impact on resistance?
Yes, a positive one. Since vaccine serotypes were often more resistant, PCV use led to substantial declines in drug-resistant pneumococcal disease overall.
What about using PCV in adults?
PCV also prevents infection in HIV plus adults. In the U.S., PCV13 followed by PPSV23 is recommended for all immunocompromised individuals. For healthy adults age 65, the 2019 update advises shared decision-making with a doctor about adding PCV13 to PPSV23, and newer, higher valency vaccines like PCV15 and PCV20 were licensed in 2021.
It's a fantastic news, extending the reach of protection even further. Beyond vaccines, what other prevention strategies are crucial in this ongoing battle?
Other prevention strategies are vital. Preventing predisposing illnesses helps so, smoking cessation, flu vaccination, better management of chronic conditions like diabetes, HIV, heart-lung disease, and fundamentally reducing antibiotic misuse. Resistance perpetuates transmission, so responsible use is key prevention.
That brings us to a crucial global perspective on prevention. How does this all play out worldwide?
Indeed. Connecting to the global health picture, the disparities are stark. Highest disease rates are in sub-Saharan Africa and southern Asia, linked to risk factors and limited healthcare access.
Right.
Despite serotype variation, a small number cause most disease universally without vaccines, making vaccination globally relevant. So disease reduction hinges on infant immunization programs, timely treatment, and reducing risk factors.
But access is an issue.
A huge issue. The stark reality is adult pneumococcal vaccines, especially for the elderly, are largely restricted to high-income countries, virtually unavailable in low-income countries where most disease occurs, a major gap.
We've taken an incredibly detailed journey through the world of pneumococcal infections, from their surprising discovery and intricate mechanisms to how our bodies fight back and the ongoing global efforts to combat them, particularly through vaccines and wise antibiotic use. You really should now have a deep, professional-level understanding of this topic.
Yeah, and if we connect this to the bigger picture, it just highlights that the fight against infectious diseases is never static, is it? We've seen how a single bacterium like S. pneumonia can evolve, adapt its identity with its capsule, and continually challenge our best treatments.
The resistance, the serotype replacement.
Exactly. It illustrates this constant microbial ingenuity, which, raises an important question. In a world where microbes constantly adapt and our interventions change their landscape, what new challenges and surprising evolutionary shifts might the pneumococcus present next? And how might continued deep research into its biology and our immune response shape our future defenses?
That's a powerful thought to ponder. The sheer adaptability of these organisms demands our constant attention and ingenuity. We hope this deep dive has given you not just facts, but a profound understanding of this intricate and ongoing battle between humanity and the microbial world. Keep exploring, keep questioning, and keep learning.